right. Good morning, everybody. So today we're going to talk about the new stuff, the cholesterol metabolism, cholesterol synthesis, and lipoproteins. Cholesterol, we know the present in the body in two forms, right? Free cholesterol and cholesterol esters. And cholesterol, of course, have two sides, good side and bad side, right? Good side means that our body needs cholesterol for making the structure of the plasma membrane or cell membrane. So cell membrane, we know, contains the phospholipids and also free cholesterol. And also cholesterol is the important precursors for the synthesis of steroids. For example, the glucocorticoid hormones and sex hormones and bile acid and vitamins. All these molecules are synthesized from cholesterol, right? So cholesterol, we need cholesterol for structure of membrane, cell membrane, and also for the synthesis of steroids, right? And bad side, we need cholesterol, but just in the right amount. If we have too much cholesterol accumulate in the tissues, peripheral tissue, particularly in like macrophages and artery walls, and then will cause some problems, right? One of them, the kind of famous problem is atherosclerosis, right? Kind of related to the cholesterol or accumulation. And the other important thing is it's the cholesterol is the major constituent of gallstones, right? So too much cholesterol definitely is not good for our health. So the cholesterol present in the tissues and also in the plasma, the circulation, and half of the cholesterol in our body from the diet, and the other half, about 50%, cholesterol actually are synthesized in the body, right? So our body is also able to synthesize and contribute to half of the total cholesterol in the body, and the other half the cholesterol actually are from the diet. So the biosynthesis, about 10% of the total cholesterol synthesized in the body actually is from the liver, right? So liver synthesized cholesterol contribute 10% of the synthesized cholesterol in the body, and also intestine, right? Liver and intestine. And all the nucleated cells are able to synthesize cholesterol, right? All the nucleated cells in the body are able to synthesize cholesterol. And in terms of the location inside of cells, where cholesterol is synthesized in the cells, that is in the ER and the cytosol, the ER facing the cytosol, right? So the closer to the ER, but still in the cytosol. And the synthesized cholesterol is synthesized from acetyl-CoA molecules. Again, acetyl-CoA is very important substrate molecules that we mentioned many times, right? And again, as acetyl-CoA is also the starting molecule for the cholesterol synthesis. And cholesterol, we know is present in the tissue and also in the circulation. So that means that some places that synthesize a lot of cholesterol, for example, the liver, that needs to be all, we absorbed a lot of cholesterol from the diet in the small intestine, and we need the transport of cholesterol from this tissue to the other tissues, right? So the transporter of cholesterol is also important in terms of the regulation of the homeostasis. And the two important lipoproteins that we're going to talk about later are responsible for the transport of cholesterol, LDL and HDL. So LDL basically is responsible for the uptake of cholesterol or cholesterol esters into the cells, into the peripheral cells, right? And HDL is a lipoprotein that transport the free cholesterol from peripheral tissue to the liver and for the excretion, to get rid of the cholesterol, right? So these two lipoproteins have the different functions in terms of the transport, cholesterol from which direction, and to deposit cholesterol in the peripheral tissue, or the dispose, cholesterol, remove cholesterol from the peripheral tissue and dispose cholesterol, right? So again, these two different lipoprotein plays a different role in terms of the cholesterol homeostasis. Now let's talk about a synthesis of a cholesterol. So how synthesized? We know that half of the cholesterol, right, in the body are synthesized in the cells in our body, and we know that acetyl-CoA is the precursors of cholesterol synthesis, right? So how cholesterol is synthesized from acetyl-CoA molecules? There are actually many steps, and I don't think you guys need to remember each steps, just kind of brief, so I just divided this whole process into several steps. I think 5 steps, and you need to know, first of all, is substrate. What is the substrate for cholesterol synthesis? Which is acetyl-CoA, right? And then the key enzymes that regulates the cholesterol synthesis, which is HSL-CoA reductase, right? So let's talk about the first step. So first step is the synthesis of mevalonate from 3 molecules of acetyl-CoA. So you guys probably familiar with this process, right? Remember when we talk about the ketone body synthesis, and the first 2 reactions actually are the same, are the common for both ketone body synthesis and cholesterol synthesis, right? So start with the 2 acetyl-CoA molecules synthesized form the acetyl-CoA, right? So acetyl-CoA is formed from 2 acetyl-CoA molecules by this enzyme called sialase, right? And then the third acetyl-CoA molecule is added to this acetyl-CoA, sorry, kind of a little bit difficult to pronounce this. So, a third acetyl-CoA molecules together with acetyl-CoA and form the 3-hydroxyl 3-methyl-urate-CoA or HMG-CoA molecules, right? And these 2 reactions, again, are the common process reactions for both ketone body synthesis and the cholesterol synthesis. And then the next reaction is the HMG-CoA is reduced to mevalonate, right? And this is the key enzyme here, HMG-CoA reductase, right? Catalyzes reduction reaction and for the formation of mevalonate, right, molecules. So again, HMG-CoA reductase is the key enzyme that control biosynthesis of cholesterol. And this enzyme can be regulated by a lot of substrates and hormones. And here you can look at here the cholesterol and mevalonate are the 2 important substrates that can inhibit HMG-CoA reductase. Bile acid is also be able to inhibit the HMG-CoA reductase. So which makes sense, right? If we have already high levels of cholesterol or mevalonate, that indicates that you have a higher rate of cholesterol synthesis, right? At this point, that we probably we have a self-regulate mechanism, right? We wanted to reduce slow down the synthesis pathway. So again, the high levels of cholesterol or veronate can inhibit HMG-CoA reductase and slow down the biosynthesis of cholesterol, right? So again, here I just wanted you to know that substrate, the acetyl-CoA, and the key enzyme into the first step, HMG-CoA reductase, and that the first two reactions are the similar or the common reaction for both ketone body synthesis and cholesterol synthesis, right? And cholesterol and veronate, that substrate can negatively feedback inhibits the HMG-CoA reductase to control the synthesis. And also the drug, the statin that people use to reduce the cholesterol levels is also HMG-CoA reductase is target of the statin. Statin can inhibit this key enzyme, right, for the cholesterol synthesis. All right, so then step two, once meveronate is formed and then will undergo 3 sequential phosphorylation and 1 decarboxylation reactions and converts to sort of isopentenyl diphosphate, right? So, and this, again, the step 2 involves the 4 enzymes, 3 kinase, and 1 diphosphate meveronate decarboxylase, right? 3 kinase that catalyze the phosphorylation, 3 steps of phosphorylation of a meveronate, like meveronate kinase, phosphomeveronate kinase, and a diphosphomeveronate kinase, right? So 4 enzymes involved this second step. And step 3 is the formation of a sequeling from isopentenyl diphosphate. And this involves isomerization, condensation, and reduction. I'm not going to go into detail, but just give you some idea. I form the sequeling. Okay, the step 4 is the sequeling. It converts to lanosterol, and the 2 enzymes that involved in this conversion, right? Okay, so this is the last step, step 5, is formation of cholesterol from lanosterol, and involve the 2 enzymes, isomerase and delta 24 reductase, and also utilize the NADPH, get the proton and to to form the cholesterol molecules. So 5 steps, and this sort of the overall process for the cholesterol synthesis start with the acetyl-CoA molecules and involve 17 enzymes, and you utilize the 8 NADPH and 3 ATPs and to form cholesterol molecules, right? But again, for the cholesterol synthesis, what you need to know is the substrate, acetyl-CoA, right? Start from acetyl-CoA molecules and a key enzyme, HMG-CoA reductase, and how HMG-CoA reductase is regulated to control the rate of cholesterol synthesis. And 2 important, the substrate cholesterol levels and the meveronate levels are very important, the negative regulators of HMG-CoA reductase, right? And also bile acid and the drug statin, and also inhibits the HMG-CoA reductase. Any questions about the whole process? Yes. You mean the acetyl-CoA, 3 acetyl-CoA molecules, right? Okay, yeah, the key enzyme is HMG-CoA reductase. So now regulation. So cholesterol homeostasis is very important, as we mentioned. We need the cholesterol for some important functions, but we don't need too much cholesterol, right? Too much cholesterol causes a lot of health problems. So that means we need to keep the balance, between cholesterol synthesis and disposal. When we need it, we synthesize. When we don't need it, and we need to dispose it, right? So the keep balance, so it's called the homeostatic regulation. So two levels of regulation. First of all is the regulation of synthesis, right? Cholesterol synthesis. When do we need cholesterol? When do we need synthesized cholesterol? And also the disposal regulation, cholesterol uptake. So remember the cholesterol present in the circulation and also the tissues. So the regulation, the uptake of cholesterol is very important. When, for example, when tissues need cholesterol and need to uptake cholesterol from circulation into the peripheral tissue, right? And when we have enough cholesterol in the peripheral tissue, and we don't want to build up cholesterol in the peripheral tissue, then we need transport cholesterol from peripheral tissue to the liver, and then to synthesize bile acid and then get excreted, right, in the testing. So this kind of regulation is very important. So so synthesis of regulation is relatively simple, but the uptake and transport and excretion, the disposal of cholesterol is a little bit complicated. But let's talk about synthesis. So synthesis of cholesterol, and again, this is a key enzyme, right? And so can be regulated by hormones and other substrates. For example, we already mentioned this: the cholesterol and mevalonate, and glucagon, and glucocorticoids, statin and starvation, or negatively regulates or inhibits HMG-CoA reductase. And insulin and thyroid hormone is a positive regulator, meaning that stimulate the HMG-CoA reductase, right? So it's relatively simple, just regulating the key enzyme of the synthesis, cholesterol synthesis. And just get a little bit more details and how cholesterol and mevalonate regulates the HMG-CoA reductase through the transcription regulation mechanisms, right? So I want you to remember this key transcription factors called SREBP, or steroid regulatory element binding protein. And this is the transcription factors. It can bind the it is called a steroid SRE. SRE is a steroid regulatory element DNA sequence. Right, so here I just represent the gene, the HMG-CoA reductase gene, and in front of gene there is a promoter, right? The promoter usually contains kind of transcription factor binding site, and so the transcription factor here, SREBP, is able to bind SRE. SRE is a DNA sequence, located the promoter of the HMG-CoA reductase gene. So once SREBP binds SRE, then can promote the transcription of HMG-CoA reductase gene. So regulate the gene expression of the HMG-CoA reductase to increase the transcription, right? So what cholesterol and mevalonate do is to inhibit the SREBP bind to SRE, so then inhibits transcription of HMG-CoA reductase gene, right? So again, this just to remember, this is SREBP is the target of cholesterol or mevalonate, and to inhibits this SREBP transcription activities and inhibits transcription of HMG-CoA reductase, right? Any questions about this process? Okay. All right. So now let's talk about lipoprotein first in order to understand better the homeostatic regulation of cholesterol, right? Because that transport of cholesterol involves the lipoprotein, right? So let's talk about lipoprotein first, and then we'll get to the regulation of a cholesterol homeostasis, right? So lipoprotein can be classified based on their densities. We actually have let me see how many, 1, 2, 3, 4, 5. 5 different lipoproteins are present in our body, in circulation particularly. And the lipoprotein has a different size, right? And also the density based on the composition of lipids versus the proteins. So if you have so for example, the chylomicron is actually the largest lipoprotein, and VLDL is second largest, right? The lipoprotein, and IDL and LDL, HLDL relatively small compared to chylomicron and VLDL. And the second important thing is that the different lipoproteins and synthesized in different places, right? For example, chylomicron is synthesized in the small intestine, right? We already talked about it, and VLDL is synthesized in the liver mainly. And the third thing is that the different lipoprotein play a different roles in terms of the transport of lipids, right? So chylomicron and VLDL are considered as the triglyceride-rich lipoproteins. That means this chylomicron and VLDL contain largely triglyceride, and the main function of chylomicron and VLDL is to transport a triglyceride, right, between the tissues. And LDL and HDL are the cholesterol-rich lipoproteins. Mean these two lipoproteins mainly play a role in the regulation of cholesterol trafficking or transport between tissues, right? And LDL, HDL are actually most abundant in the plasma. The reason is because these small lipoproteins relatively stable compared to chylomicron and VLDL, so degraded kind of slowly compared to chylomicron and VLDL. So that's why they stay in the plasma longer, right? So that makes, they are more abundant compared to VLDL and chylomicron. All right, so now let's look at the composition of a lipoprotein. So in general, 5 different lipoproteins all compose the lipid core, which contain the triglyceride and the cholesterol ester. Remember, cholesterol ester in the core, right? The lipid in the core are the triglyceride and the cholesterol ester, right? Not free cholesterol. And also the surface monolayer of phospholipids, free cholesterol, and structure proteins, also called apolipoproteins or apoproteins, right? So mainly the lipoproteins contain the lipid core and the surface layer, right? So surface layer contains the monolayer of phospholipids, right? And the free cholesterol. Free cholesterol usually located on the surface, right? And the structure proteins or apoproteins, right? Apoproteins also localized on the surface of the lipoproteins. So here give you maybe a little bit better view of the lipoprotein. How lipoprotein looks like, right? In like a ball, right? Inside of the ball is the called lipid core. Again, it contains triglyceride and cholesterol esters, and the surface, the phospholipids, single layer phospholipids, and free cholesterol, and a different type of apoproteins, apolipoproteins, right? So different type of lipoproteins, they contain different composition of the lipid cores, lipids in the core, and also different apoproteins, right? So now let's talk about apoprotein. So what is what kind of apoprotein that localize on the surface of the lipoproteins? That in general we have apoprotein A, B, and different A, type A and B, like B40 and B100, C, C1, C2, and C3, and ApoE, right? So a variety of apoproteins that are present on the surface of the lipoproteins. Again, not every type of apoprotein are needed for the for the different type of lipoproteins. And what is a function of the lipoprotein? Why we need the lipoprotein? So first, some of the lipoproteins that serve sort of as the cofactors or enzymes. For example, ApoC2. So try to remember this, right? ApoC2 is the apoprotein that serve as a cofactors of enzyme, particularly for lipoprotein lipase. LPL, remember LPL. So what does LPL do? What does LPL do? Remember? Hydrolyze the triglyceride, right? Remember, in both, in which the lipoproteins. Remember? Yeah, where? Right? Right, that correct. I mean the triglyceride, where is the triglyceride located? That it's hydrolyzed by LPL. Yeah, VLDL and the chylomicron, right? So LPL hydrolyze the triglyceride in the VLDL and the chylomicron, right? So that makes sense, right? So you connect this, ApoC2. So you will expect chylomicron and VLDL should have ApoC2 on their surface, right? So then ApoC2 that help LPL to hydrolyze triglyceride in the chylomicron and VLDL, right? So this is one important function of apoprotein. And the second is the apoprotein serve as a structure integrity, to stabilize lipoproteins. And third, some of the apoprotein is also very important, serve the ligand for receptor binding. We're going to talk about later. Try to remember this, right? ApoE and ApoB100 are very important lipoprotein that on the surface of the the lipoprotein, particularly LDL, and that help the LDL lipoprotein to bind its receptor, LDL receptor or LDL receptor, LDL related protein. Then an LDL receptor or LRP usually are located on the surface of the tissue cells like liver and other peripheral cells. And so ApoE2, ApoE and ApoB100 on the surface of a lipoprotein can help lipoprotein bind the receptor on the cell surface and then, uptake by the cells, right? So this is also one of the important functions of apoproteins. And lastly, apoproteins also help suspension of the lipids in the plasma, right? So again, these are the very important, the two ApoC2 and ApoE and ApoB100, how they help the, the LPL hydrolysis and the uptake of LDL. Okay, so this table I just give you a overview of the 5 different types of lipoproteins and what are the core lipids and surface lipids and apoproteins on each apoproteins. So I mentioned before that chylomicron and VLDL are the class triglyceride-rich lipoprotein, right? So you can see in the core, the chylomicron had 85 to 95% of lipids in the core are triglyceride, right? And 55 to 65, the lipids are triglyceride in the VLDL. So the majority of the lipids in the core in chylomicron and VLDL are triglyceride, right? And also have a relatively small percentage of lipids in the core are the cholesterol esters in the VLDL chylomicron, right? So that's why these two lipoproteins are called triglyceride-rich lipoproteins, and you can also imagine the function of these two lipoproteins are to transport triglyceride, right? And also try to remember the apoproteins on the membranes of chylomicron and VLDL. And these two ApoB are different between chylomicron and VLDL, right? So chylomicron, the ApoB48 is synthesized in the small intestine, right? So that's why ApoB48 usually is packed into the chylomicron, right? So chylomicron usually contains the ApoB48, which is synthesized in the small intestine, which makes sense why we connect with the synthesis of chylomicron in the small intestine. And VLDL, ApoB100 is a major ApoB protein on the surface of a VLDL, which also makes sense, right? ApoB100 is synthesized in the liver, and VLDL is also synthesized in the liver. And in addition to ApoB, either 48 or 100, and these two lipoproteins also contain ApoC2 and C3 and ApoE. So ApoC2 we also mentioned, right? And it's very important to help. It's cofactors of LPL, right? To help LPL function. So it's LPL. We know that hydrolyzed triglyceride in these two lipoproteins, right? So it probably help you to memorize, this ApoC2 that connected to the lipoprotein lipase and the chylomicron and VLDL, right? Does that make sense? Okay. And the LDL and HDL, you can see, the cholesterol-rich lipoproteins. So you can see that contain more cholesterol esters than triglyceride in LDL and HDL, right? So again, the function of LDL and HDL is to regulate or transport the cholesterol. And LDL and pretty simple, only have ApoB100, right? Or the major apoproteins, ApoB100. And HDL contains ApoA1 and ApoA2, and IDL is ApoB100 and ApoE, right? So, any questions about the composition of the lipoprotein? Okay, so and the other important concept is called a remnant of a lipoprotein. That means that the lipoprotein, the composition, not always stable, means that they actually change, right? You know, depend on the how much the lipids then be removed from lipoproteins. So again, the size or composition of lipoproteins is changing. Right? So for example, chylomicron, once it's synthesized in the small intestine and contains a lot of triglyceride, right? And then chylomicron being transported to the peripheral tissue, for example, particularly adipose tissue, right? And then LPL, lipoprotein lipase, the hydrolyzed triglyceride inside of the chylomicron and release fatty acids and pick up by adipose tissue, right? So that means once it's reached the adipose tissue, there are a lot of triglyceride being hydrolyzed and pick up into the adipose tissue. So then chylomicron actually lose the triglyceride, right? So triglyceride pick up by the peripheral tissue, particularly adipose tissue. So then the chylomicron and become the chylomicron remnant. And chylomicron remnant finally is picked up by the liver. So chylomicron remnant, if you compare chylomicron remnant to chylomicron, the size is smaller and the content of a triglyceride is much less, right? Because you know it's being uptaken by the adipose tissue. So and similar for the VLDL is synthesized in the liver and also goes to the peripheral tissue, including adipose tissue and muscle as well. So and also triglyceride in the VLDL and pick up by the adipose tissue and muscle, and you're also losing the triglyceride. And so VLDL eventually become to IDL or LDL, right? So LDL basically is kind of a VLDL remnant, right? So once the triglyceride being picked up by peripheral tissue, then the VLDL have less triglyceride but more cholesterol esters. So there is also kind of called the exchange of lipids between the LDL, IDL, and HDL. So I don't want to make too complicated here, but just wanted to mention the exchange lipids between HDL and IDL or LDL that makes LDL more cholesterol rich, means that the LDL can receive the cholesterol esters from HDL that become, you know, cholesterol rich lipoproteins, right? But we will get there later, right, in more details. All right. So, any questions about the remnant lipoprotein? Right. Yeah. So, what is the difference between chylomicron and chylomicron remnant in terms of a lipid composition? We already mentioned, right? Chylomicron have a more triglyceride and bigger compared to the chylomicron remnant, right? Chylomicron remnant has less triglyceride. Okay, so now let's talk about individual chylomicron. What is the composition and the function? Where the chylomicron synthesized, right? So the first chylomicron, which is the largest lipoproteins in the circulation, is the chylomicron. So chylomicron, we know the core lipids in the chylomicron contains triglyceride and cholesterol, right? But more triglyceride than cholesterol esters. And apoproteins. What type of apoprotein is on the chylomicron surface? Do you guys remember the table that I showed you? ApoB48, right? Yes. So ApoB48 is the apoprotein that's synthesized in the small intestine, and also ApoA1, 2, 4, ApoC2, right, and 3, and ApoE. So I just want you guys try to remember these two important apoproteins on the surface of chylomicron, ApoB48 and ApoC2, right? The ApoB48, because it's unique, it's synthesized in the small intestine, right? It's on the chylomicron but not on the VLDL. And ApoC2 is because it's also important function, right, to help LPL, hydrolyzed triglyceride. So try to remember these two apoproteins on the chylomicron surface. And synthesis. So where are chylomicron synthesized? Small intestine, right? Small intestine enterocytes is the place where chylomicron synthesized. So where does triglyceride in chylomicron come from? Diet, yes. So from diet, because it's synthesized in the small intestine, right? So basically, our body gets the lipids and packed into chylomicron and transported into the adipose tissue. And ApoB48 is made from enterocytes, right? Okay. Now let's talk about the metabolic fate of chylomicron, how chylomicrons metabolize, and what is the function of chylomicrons. Right? So first, the function of chylomicron is to transport triglyceride or dietary triglyceride, right, from small intestine to the peripheral tissue, particularly adipose tissue, right? Now, how chylomicron is metabolized in the body? So first, the chylomicron is made from the small intestine, right? And packed, so triglyceride absorbed from the diet, then packed into the chylomicron. And the chylomicron, when the first synthesized, is called a nascent chylomicron. That means the chylomicron, this chylomicron does not have the full range of the apoprotein, right? So you can see the nascent chylomicron has ApoB48 because it's also synthesized in the small intestine, right? And also ApoA, but does not have ApoC and ApoE yet. And then the ApoC, ApoE basically is transferred from HDL to nascent chylomicron, right? And finally, chylomicron receives the ApoC, ApoE from HDL and get all of the apoproteins on the cell surface. So then become the mature chylomicron, right? So mature chylomicron is very important, particularly the C, the ApoC2, right? So mature chylomicron has the ApoC2, and that is very important for the LPL hydrolysis, right? So once the mature chylomicron reach the peripheral tissue, particularly adipose tissue, and adipose tissue release the lipoprotein LPL, right? So, and this LPL can hydrolyze triglyceride in the chylomicron and with the assistance, of the ApoC2, then release fatty acids and glycerol. So fatty acids can be taken up by tissue, adipose tissue, and used for the synthesized triglyceride in the fat condition, right? In the adipose tissue. And then chylomicron becomes chylomicron remnant. So you can see the size becomes smaller, right? Because a lot of triglyceride being being breakdown and uptake by adipose tissue and other tissue, right? So chylomicron remnant becomes smaller and have a less triglyceride compared to mature chylomicron. And also the ApoC, ApoA is go back to the HDL, right? So you see so chylomicron remnant does not have a ApoC. ApoC only ApoB 48 and ApoE. And because the chylomicron remnant eventually also pick up by the liver, so liver cells on the cell surface of liver has a LDL receptor and LRP receptors, right? Has both receptors. And these receptors can recognize the very important lipoprotein, remember lipoprotein, apoprotein E or apoprotein B100, right? So once the chylomicron remnant has the ApoE, then can bind receptors, right? And then pick up by the liver. So triglyceride in the chylomicron remnants eventually hydrolyzed in the liver by a different enzyme called hepatic lipase, right? So hepatic lipase hydrolyzes triglyceride in the chylomicron remnant. And again, important, ApoE is very is a key apoprotein that help the chylomicron bind to this receptor and for the uptake. Right? Any questions about this process? All right. So let's take a 10 minutes break. Okay, let's move on. So, chylomicron, right? We talk about the where that is chylomicron synthesized from small intestine, and the main function of the chylomicron is to transport the triglyceride, which is initially absorbed from the diet from small intestine to the peripheral tissue, particularly in adipose tissue. And once it's synthesized, initially it's not, it's like the nascent chylomicron does not have a other two important apoproteins, particularly ApoC, ApoC2, and ApoE. And once this chylomicron get these two apoproteins from HDL and then become mature chylomicron. And again, the ApoC2 is very important to promote the hydrolysis of triglyceride by LPL. And once triglyceride hydrolyzed and picked up by adipose tissue and other tissue, then chylomicron size becomes smaller and lose a lot of triglyceride and become chylomicron remnant. And ApoE is important, the apoprotein that can help the chylomicron remnant bind to the LDL receptors or LRP receptor for pickup by the liver, right? And again, triglyceride in the chylomicron remnant, and eventually hydrolyzed by hepatic lipase in the liver. Okay, so let's just give you sort of an overview of the chylomicron metabolism. Again, synthesize chylomicron, become mature, and then transported triglyceride into the adipose tissue, and then lose about 90% of triglyceride once it's reached the adipose tissue and other peripheral tissue. Then also lose the ApoA and ApoC, and then become chylomicron remnant. So chylomicron remnant eventually pick up by the liver through this receptor mechanism, and ApoE is important, right, for the binding of the chylomicron remnant to the LDL receptors. So, and the function is here, a lot of triglyceride in the chylomicron initially from the diet, right, from the small intestine being picked up by the tissue, right, adipose tissue and other tissue. So this is the main function of chylomicron, is to, again, to transport the triglyceride from the diet and to the adipose tissue and other tissue, right? This whole process usually takes place in the fat condition, right? Only in the fat condition we have a lot of triglyceride absorbed from diet, and then chylomicron is synthesized and transport the triglyceride to the adipose tissue for storage, and also liver, right? Chylomicron remnant. Okay, so here's a question. So which enzyme hydrolyze triglyceride in chylomicron? LPL, right? LPL that's released from adipose tissue. And which enzyme hydrolyze triglyceride in chylomicron remnant? Hepatic lipase, right? And hepatic lipase, by its name, you know that it's synthesized or located in the liver, right? So liver also has its lipase. Okay, now let's talk about VLDL. So core lipids in VLDL. What are the core lipids in the VLDL? What is the major lipids? triglyceride, right? So triglyceride again is also the major lipids in the VLDL in the core, and also has the relatively small percentage of cholesterol esters and apoproteins in the VLDL, which ApoB or ApoB 100, yes, is synthesized in the liver, right? So ApoB 100 is unique ApoB proteins in the VLDL, right? Because it's synthesized from in the liver, and also VLDL has a ApoC2, which is also important, right? It's you need LPL need ApoC2 for hydrolyzing the triglyceride in VLDL also, and ApoE. And synthesis. So where are VLDL synthesized? Liver, right? In the liver. So VLDL is synthesized in the liver. And where do the triglyceride in VLDL come from? Where the triglyceride in the VLDL comes from? From three sources. First is de novo synthesis from the dietary carbohydrate. Remember lipogenesis, we talk about lipogenesis, right? In the fat condition, particularly when we eat a high carbohydrate diet, so excess carbohydrate can be converted to fatty acids and then synthesize triglyceride. So this is an important sources of triglyceride in the VLDL. And the second is the long-chain fatty acids internalized by the liver from plasma lipoprotein remnant. For example, chylomicron in the fat condition, right? Chylomicron remnant. Also, liver picks up the triglyceride in chylomicron remnant. So this also is a sources of the triglyceride. So chylomicron remnant arrive in the liver, then hepatic lipase hydrolyze triglyceride in chylomicron remnant and release fatty acids, right? And fatty acids then be re-acidified to triglyceride and repacked into the VLDL, right? So and this is the second source of the triglyceride. And lastly, the unacidified or free fatty acids. So sometimes when, and the third sources of the triglyceride that actually happen during the fasting. So during the fasting, our body have a lot of fatty acids being released from adipose tissue, right? Through the adipose tissue, through the triglyceride mobilization. And so then a lot of fatty acids into the circulation that can be picked up by the liver and re-acidified to triglyceride and packed into the VLDL. And under the fasting condition, the VLDL also released, but mostly picked up by the muscle, right? And triglyceride break down fatty acids and pick up by muscle and utilize, as an alternative energy during the fasting. So the first two sources of triglyceride packed into VLDL for VLDL synthesis mostly occur in the fat condition, right? So in the fat condition, triglyceride in VLDL mostly comes from lipogenesis, from carbohydrate, right? And from the chylomicron remnant, which initially from the diet. Does that make sense, right? And the third source during the basically is contribute free fatty acids, raise the free fatty acids level, contributing to the triglyceride in the VLDL, basically occur in the fasting condition. And also the final destination of the VLDL is also different between the fasting and the fat condition. So in the fat condition, most of VLDL being transported into adipose tissue, right, and for storage, the triglyceride in there. But during the fasting, the VLDL usually is go to the muscle and deliver fatty acids to muscle for energy. And ApoB 100 is synthesized in the liver. Right? This is a unique ApoB for the VLDL 100. All right, any questions? All right, so metabolic fate of VLDL, how VLDL is metabolized. And again, also similar question, what is the function of VLDL? I just described, right? Depend on the physiological condition, the fat condition, that VLDL transported, the triglyceride basically from the lipogenesis, right, in the liver, it is converted from carbohydrate, and from the diet, from carbohydrate remnant, right, this triglyceride initially from the diet, so, and transported the triglyceride to adipose tissue for storage. And during the fasting condition, that the triglyceride basically is from the circulating fatty acids, right? This fatty acids initially from the adipose tissue, right, break down triglyceride. And in the fasting, VLDL transported triglyceride to the muscle for breaking down fatty acids and utilized as energy in the muscle. So this is just the general, picture of the VLDL, how VLDL functioning in transport the triglyceride. Again, depend on the physiological condition. And so here is the whole process. So again, the VLDL, once it's synthesized, also in the form called a nascent VLDL, because it also lacks the ApoC and ApoE. And again, the nascent VLDL receives ApoC and ApoE from HDL and become mature VLDL. And again, destination or peripheral tissue can be adipose tissue or can be skeletal muscle or hard muscle, depend on the physiological condition, right? So in the fat condition, destination should be adipose tissue, and similar to the chylomicron, the C ApoC2 is also on the surface of VLDL, and it is also important for LPL, hydrolyzed triglyceride, in the VLDL, right? So in the fat condition, the fatty acids be picked up by adipose tissue and re-acidified triglyceride and stored in the adipose tissue. But in the fasting condition, the fatty acids actually goes into the muscle and for oxidize, right, for oxidation and energy production. And once this triglyceride being picked up by the peripheral tissue and the VLDL becomes remnant, so IDL, right? So IDL is, you can see also, size is much smaller because it loses a lot of triglyceride, right? And it's also ApoC is also lost and go back to the HDL, and where you still have ApoE remain because it's an ApoB100, and these two apoproteins is very important for pickup by receptors, LDL receptors, right? And also I mentioned before that there is the exchange of lipids between LDL and also actually IDL. L here. So between HDL and LDL or IDL, that means that the triglyceride is given to the HDL, and the cholesterol being transferred from HDL to the LDL. So eventually LDL become the more cholesterol ester rich, right? And and less triglyceride. And LDL can be picked up by liver and also other peripheral tissues, for example, macrophages and in artery wall. This is very important for the development of the atherosclerosis. We're going to talk about later. So this is the whole process of the the VLDL metabolism, right? So and you also can see the relationship between the VLDL and LDL, right? And also there is lipid exchange happening between HDL and LDL. So that makes the composition of lipids change after the exchange of lipids, right? So the VLDL remnant eventually becomes the LDL. Right? Any questions? Right? Okay, so again, this is a just overall picture of the VLDL metabolism synthesized in the liver, right? And packed triglyceride can be from 3 different sources, right? Depend on the again physiologic condition, fasting condition, and the purpose of the VLDL is to transport the triglyceride to the tissues, and once the triglyceride being picked up by the tissues, then VLDL become remnant. You have lots of triglyceride is lost, and also some the ApoC is also being transferred back to the HDL, and they are also exchanged between the IDL and HDL, and the more triglyceride being moved to HDL and the cholesterol esters being added to the IDL, so eventually IDL become the cholesterol-rich LDL, right? So again, this will LDL will be picked up. Both IDL also can be picked up, right? Both IDL and LDL being picked up by the liver or other peripheral tissues, and the important the apoprotein that mediate the pickup is ApoB100 and ApoE. Okay. All right. So now let's go back to the LPL. We talked about it very early, and also we mentioned here, and that is very now. I think we have a closer connection, to the lipoprotein, right? So LPL, we know main function is to hydrolyze triglyceride in chylomicron and VLDL, and synthesized mainly in just try to remember, fresh your mind again, the 2 main tissues, right? Remember, adipose tissue and the muscle, and located on the wall of capillary endothelium, and within the tissue, in adipose tissue, muscle, including skeletal muscle and heart muscle. But LPL is not in the liver. Liver has its own lipase. It's called a hepatic lipase, right? And the function of this LPL enzyme is to hydrolyze triglyceride in chylomicron and VLDL to release fatty acids, and this fatty acid can be transported into tissue. Again, depending on which tissue release LPL and in which physiologic condition, and have a different activities in different tissues. For example, have a lower Km for triglyceride in the muscle and high Km for triglyceride in the adipose tissue. And during the fasting, the LPL activity is higher in the which tissue? muscle, right? Should be higher in muscle, and in the fat condition, the LPL activity should be higher in the adipose tissue. And again, this is a new concept that we just talked about today. Is the LPL activity requires the presence of ApoC2, right? So ApoC2 again is very important to facilitate the functional activities of LPL. And insulin stimulates LPL synthesis and secretion from which tissues? The adipose tissue, right? Okay, so here I have a question. So how are triglycerides in LDL and chylomicron metabolized? It's kind of a big question, right? You guys, we already described the process, how VLDL and chylomicron metabolize. And which enzyme is the key for VLDL and chylomicron triglyceride metabolism? Which enzyme mean to hydrolyze triglyceride? Yeah, lipoprotein lipase, LPL. Right? Just try to remember this. And the ApoC2 is important for this enzyme activities, right? Okay, let's see. Let me just see if I have more. Okay, so there is just more information about the LPL activity in skeletal muscle. So in the fasted state or exercise, this just pretty much the diagram pretty much just describes that I already talked about it. In the fat condition, the fasted condition or exercise, catecholamine levels increase, right? So then can break down the triglyceride in the adipose tissue and release fatty acids. So raise the blood circulating the circulating levels of the free fatty acids, and fatty acids are picked up by the liver and synthesize triglyceride, and is packed into the VLDL. And this VLDL mostly is travel to the skeletal muscle, right? And this is the LPL that's synthesized from the muscle during fasting or exercise. So muscle synthesize more LPL released and hydrolyze triglyceride in VLDL and break down fatty acids. And this fatty acids is picked up by the muscle usually for oxidize, right? For oxidation and ATP production energy. So this is the muscle. And let's see. Okay, so let's just talk about the cholesterol metabolism in the LDL and HDL. Right? We finished the triglyceride metabolism that mediated by two major triglyceride-rich lipoprotein, chylomicron and VLDL. And here we switch to the gear to the cholesterol metabolism, right? And this is mediated by the cholesterol-rich LDL and HDL. It should be HDL. I don't know why I said LDL. So usually people divided the cholesterol as the bad and good cholesterol, right? So bad cholesterol, for example, the LDL, particularly LDL, is considered the bad cholesterol, and the good cholesterol, HDL, right? And so the main reason is because LDL usually is located in the peripheral tissue that, the LDL receptor located in the peripheral tissue, for example, macrophages that can pick up the circulating LDL cholesterol and deposit the cholesterol in the peripheral tissue, right? And the HDL considered is good because HDL usually they pick up the cholesterol from peripheral tissue and transport it to the liver for the disposal. So HDL considered is good because if we have a more HDL, then we can dispose more cholesterol, right, to prevent the accumulation of cholesterol in the peripheral tissue. So this is just the main, the different, the function of these different cholesterol-rich lipoproteins. Now let's talk about LDL first. So LDL, core lipids, of course, is the cholesterol ester will be the major lipids, right, and less triglyceride. And ApoB100 is a major lipoprotein that on the surface of LDL. And IDL is the VLDL remnant. We already know that, right? It's also have cholesterol esters and triglyceride and ApoB100, ApoE. And the metabolism, in terms of metabolism or the function of LDL, usually again I mentioned, right, the LDL receptors is localized in the peripheral cells. So the LDL basically is transport cholesterol esters into the peripheral tissue. So deposit triglyceride deposit cholesterol in the peripheral tissues, right, and pick up by this receptor-mediated process. And ApoB100E that mediates the binding of LDL, IDL to LDL receptor. This we mentioned many times, right? Again, the ApoB100, ApoE on the surface of the LDL, IDL mediate the uptake by the LDL receptors into the peripheral cells. And receptor expressed mainly in the liver, but also in the peripheral tissue cells as well. And this important concept here. So the number of LDL receptors in the liver that determine or the other peripheral tissue, and that determines the rate of internalization of the LDL and not the concentration of LDL in the plasma. Do you guys clear? Understand about this? So the receptor, LDL receptor, actually control the uptake of cholesterol, right? So cholesterol level in the circulation is not as important as the number of receptors. So if you have a lower or higher, the lower LDL receptor in the, let's say in the liver, then liver is not able to pick as much as LDL in the circulation, right? It's compared to the person that has a normal LDL receptor on the liver. So that means you have a lot of LDL that remain in the circulation, right? So you raise the LDL levels in the circulation if you have a lower number of the LDL receptor in the liver. So then these high levels of LDL will go to the peripheral tissue and pick up by peripheral tissue, peripheral cells. So you kind of shift the deposition or the the LDL from liver to the peripheral tissue, including macrophages. So that will increase the uptake of LDL by the peripheral cells, including macrophages. That will the major problem that causing the atherosclerosis. You know, you have a lot of cholesterol that deposit in the peripheral cells, right? So it's not the concentration. Concentration is not the most important. The most important is the receptor numbers, right? Does that make sense? Any questions about this concept? Okay. So now LDL receptor. So LDL receptor, let's look at this diagram. Let's represent the cell membrane, right, and the cytosol. And so the cell membrane has the LDL receptor. So LDL receptor has a binding site for ApoE and ApoB100. You have a question? Uh-huh. Yeah, that's good question. So receptor is one of the regulatory mechanism, and we're going to talk about later about the cellular regulation. You know, including you talking about the synthesize in the body. So, I think the genetic problem is also maybe including receptors and also the some you know the mechanism that involve the synthesis of the cholesterol. So genetic involve actually many aspects. One is whether this person be able to synthesize more cholesterol or less cholesterol, and the second thing is that whether this person that be able to metabolize cholesterol, including you know pick up by the liver and excreted you know cholesterol as a bile acid, or whether it's more easy to deposit you know in the peripheral cells, including all this kind of process. I mean, so so this the LDL receptor on the cell surface. So LDL binds receptor through the again ApoE and ApoB100, right? And once it's bound to LDL receptor and the whole LDL together with receptor that internalize right into the cells, and then the cholesterol then be metabolized in the cells, inside of cells, right? And this is more information about the receptor mediated, the uptake of LDL, and the metabolism of LDL. So again, this represents cell membrane and the LDL receptors, and LDL binds the receptor and internalized similarly, and then inside of cells, the cholesterol can be metabolized, and also the apoproteins can be break down to amino acids, right? And cholesterol ester being released to cholesterol. And the receptor can be recycled, right? You know, can go back to the cell surface membrane. And cholesterol-free cholesterol, once it's released from the LDL, and can be re-acidified to cholesterol ester inside of cells by this enzyme ACAT. Right? And in the cells, I just showed you the last slide, the cholesterol-free cholesterol can be re-acidified to cholesterol ester. This is a reaction of the catalyzed by ACAT, right? And then cholesterol esters can have several phases. First, it can provide substrate for synthesis of bile acid and steroid hormone, which this process occurs in the liver, right? And supplies as a colipids for chylomicron and VLDL synthesis, and also can be used for form the cholesterol ester and sperm cells, which can occur in the in the macrophages, right? And it also can be hydrolyzed by HSL to release free cholesterol, right? As needed in the cells. So ACAT is an enzyme called acyl-CoA cholesterol acyl-transferase. Right? This is an enzyme that catalyzes reacidification of a cholesterol. Okay, now HDL. So HDL and core lipids is similar to LDL, right? And cholesterol ester is a major lipids and also has some triglyceride. And apoproteins are ApoA1 and ApoA2. And in terms of the location where HDL synthesized is synthesized in the liver and intestine, and the function of HDL is very important. It's considered a good cholesterol, right? Because it's very important for cholesterol homeostasis and transport the cholesterol from peripheral tissue back to the liver, right? Try to imbalance with the transport of cholesterol from liver to the peripheral tissue, so can prevent the accumulation of unnecessary cholesterol in the peripheral tissue cells, right? So HDL is very important. So it's also called a reverse cholesterol transporter, right? Means that we transfer cholesterol from peripheral tissue back to the liver and for the disposal. So it's very important for the balance, or homeostasis of cholesterol and prevent accumulation of cholesterol in the peripheral tissue cells. And the other function is we already talk about it a lot, right? It's called a repository for ApoC, ApoE for both chylomicron and VLDL. Remember, both nascent chylomicron and VLDL need to get ApoC and ApoE from HDL, right? So HDL usually provides the ApoC and ApoE for chylomicron, for maturation of chylomicron and VLDL. And this is a concept I wanted to introduce to you, is the reverse cholesterol transporter. Means that transport cholesterol from peripheral tissue back to the liver and for the disposal. So this is the very important process, right? And this process is usually accomplished by so-called HDL cycle, right? HDL cycle. So HDL cycle means that there's two different type of HDL, alpha HDL and pre-beta HDL kind of conversion, right? Means that, and then the thing is that the alpha HDL and pre-beta HDL has a different lipid composition, right? And apoprotein composition. So means that alpha HDL can be converted to beta HDL and vice versa. So this interconversion occurs and to accomplish the reverse transport of cholesterol, right? Okay, so before we talk about the detail of HDL cycle, I want you guys to watch the video and to get a better idea, maybe a kind of in a relaxed way, right? Let's see.
Reducing atherogenic burden is one of the greatest challenges for today's physician facing the epidemic of atherosclerosis. Fortunately, research is yielding enormous insights to meet this challenge. Recent decades of study have proven that lowering LDL cholesterol reduces cardiovascular disease. Yet still, many cardiovascular events occur without significant elevation of LDL cholesterol. Newer data are revealing significant atherogenic potential in lipoprotein particles other than LDL. In some circumstances, the composition of any of the lipoprotein particles can be altered, allowing participation in the atherogenic process. Furthermore, it has been shown that hypertriglyceridemia is a marker for the existence of such atherogenic particles. Hypertriglyceridemic patients at risk exhibit elevated triglycerides and low HDL cholesterol. This profile is often associated with the metabolic syndrome and diabetes, resulting from overnutrition, obesity, and physical inactivity, and leading to premature atherosclerosis. The epidemic of metabolic syndrome is expanding the high-risk patients.
The difference between possible and impossible?
Reducing atherogenic burden is one of the greatest challenges for today's physician facing the epidemic of atherosclerosis. Fortunately, research is yielding enormous insights to meet this challenge. Recent decades of study have proven that lowering LDL cholesterol reduces cardiovascular disease. Yet still, many cardiovascular events occur without significant elevation of LDL cholesterol. Newer data are revealing significant atherogenic potential in lipoprotein particles